Welcome back to our class. Today we are going to be talking about DNA replication. Last time we talked about um, base calculation, and we saw how we can do these base calculations. So today we're going to be talking about DNA replication. When time allows, then we shall talk about protein synthesis. Why? Because you need to know these concepts so that you can know what will happen to the next topic. If you don't understand the first topic, you won't be able to understand the second topic. There are some questions we have to ask you about, and then you're supposed to go to the paper when you know these questions. And then I'm going to show you some of the questions they might ask you in the paper, and then you'll be able to answer those questions. You're going to go through them together, and then we'll be able to answer those questions. If you have any question, let me know in the comment section. I'll be able to answer those questions. Um, Number two, if the sound is not right or correctly, please let me know so that we can work upon it. All right, let's go to the class without wasting time. What is DNA replication? Some of the questions they might ask you is about what is DNA replication. When you talk about DNA replication, basically you are talking about DNA making identical copies of itself. So if, for example, I have 1, 1, 1, 1, 1, 1, yes, I will have the new DNA which is 1, 1, 1, 1, 1, 1. For example, that's it. So making identical, if this strand and this strand, or this DNA and this DNA, DNA A and DNA B must be the same. How does it become the same? We saw that we use the bases, whereby A goes with the T, and then C goes with the G. And then we saw that, for example, if you find T, then automatically that is DNA, while if you find U, that is RNA. So we're not going to talk about this. So let's go to the examination, our booklet, and then we see exactly what is going to happen. What is DNA replication? When you talk about what is DNA replication, what is DNA replication? DNA replication is the process by which DNA makes. You see, this thing is in different color, which means that it's a marking point. If you don't add this in the answer, then you won't be able to get the answer correctly. DNA makes identical copy of itself or exactly copies of itself. Yes. When does it occur? It occurs, this DNA replication only occurs during cell division. When does it, when does it occur in actual sense? We are looking at during interphase or before the cell divides. So we shall say that DNA replication occurs before the cell divides, before cell division, or during interphase. If you don't talk about interphase or before the cell divides, you won't get a mark, because that's what you are looking for. Where does it occur? It occurs in the nucleus. Look at this critically. Occurs in the nucleus. This is what you are looking for. If you don't have it, you won't be able to get that mark. It occurs in the nucleus. You have to be specific to whatever you are talking about. Please don't write too much. We're not about too much. We are about the facts. And the facts in this booklet, they are indicated by the different color, not in black, which is a marking point. All right. Why does it occur? Why should we have DNA replication? DNA replication, we have DNA replication. Why? So that we double the chromosome number. We double the chromosome number. This word double Doubling the chromosome number is very important. We double the chromosome number. Why? The reason why DNA replication occurs, look, you have a cell. You have cell A, and then now this cell A has two chromosomes. Example. Now, this cell, let me just do it like this. You have cell A, yes. Before the cell divides, it has this DNA. This DNA is supposed to split into two during the cell division, whereby this cell is going to have this cell and also is going to have this cell during cell division. But now, if this DNA is not divided into two, it means that one cell, for example, cell A, will have the DNA, while cell B will have no DNA. So, what does the cell do? It makes this DNA to be doubled. You see? So that's the function of DNA replication. So that when the cell is dividing, it can have, each cell can have equal number of genetic material. For example, now, if it was like this, so when it divides, yes, during the second phase, this part, you see, it splits. Whereby, let me just do like this. This part, let me use a different color so that you see exactly. So it's gonna split like this, whereby this chromosome will go to this cell, while this chromosome is gonna go to that cell or that part of the chromosome chromatid. So now, in case this DNA did not replicate, how will this going to get these chromatids? Once they reach the cell, they become the chromosome. That's why we shall say that DNA replication occurs. Why? To double the chromosome number, so that during the cell division, equal chromosomes can go to each what? Each cell. So basically, results in the formation of identical cells. Yes, that's what we are talking about here. That so that A and B, they can have equal number of genetic material. All right, let's continue to. Thank you, Sefiso Zulu. Let's go to the next question. How does it occur? When you talk about how does DNA replication occurs, they're asking you the process of DNA replication. Guys, don't go to the paper without knowing this. Because when you go to the paper without knowing that, it means that you are just killing yourself. There is no way, I repeat, there is no way you're gonna go to the paper without the following equations. One must come: DNA replication, transcription, translation, the structure of DNA. Well, when you know that, go when you know those four short answer questions. So, because if you don't go there, you're gonna bring one. So, if you don't do it, then you won't be able to get those 6 marks. So, guys, please don't ask for scope. The scope is always there. Yes, we always even do predictions before you write the paper, and then you almost find 99.999% of what you have predicted. So just keep yourself on Thundereduc. You'll be able to get distinctions. All right, let's continue, guys. How does it occur? But DNA is a double helix. I explain this. People confuse what is double helix and what is double stranded. Those are two different things. When you say it's double helix, you know it's helical, helicopter, helicopter, helicopter. You know that song, né? Or if you're a kid, all right. So it's this one is helical, you see. Also, this one is also helical. So it becomes double. So we shall say that this is double helix. What about now? When we say it's double stranded, we are looking at this. You see, it's strand one, strand two, strand two, strand one. So the DNA is double-stranded, meaning that it has two strands. Double helix and double-stranded, they are two different things. So if you mix them, you are lost. Yes. So they are not supposed to be you are not supposed to make them to mean the same. Double helix, that's double helix, this is double stranded. So basically, step number one, DNA double helix, if you look at it here, DNA double helix, if you look at it here, yes, if you look at it here, ne, you'll find out that this. If you look at it there, you'll find out that this is double helix. When it is double helix, it unwinds. It means that it uncoils. Don't say uncoils in a paper. You know what? The students, you have a problem. When you tell someone, don't say this, that's what he says. Yes. Although, sorry, although here I said um, unwinding. Unwinding in actual sense means uncoiling. Don't say uncoiling. It's like you have the hair on the on the head. You know, you ladies, you know, they are coiled. Before you remove them, you uncoil them. That's the meaning in science. We shall say that unwinds. Now, when it unwinds, in actual sense, after unwinding, the weak hydrogen bonds will break. In science, we shall say that it has unzipped. DNA double helix unwind, weak hydrogen bonds break. So in that actual sense, these bonds break. Let me zoom it out and then you see it clear. Yes. Ne? When these bonds break, I think now you can see clear. Yes, when these bonds break, then you are able to see exactly what happens. You see? So these bonds, they will break. When they break, we have bonds in there. In science, we shall say that it has unzipped. You understand? Weak hydrogen bonds break. Remember, these bases are being connected with three bonds there, with the two bonds there, with three bonds there, with the two bonds there, with three bonds there. You see? So those bonds, they break. When they break, what happens? When they break, we shall say that it has unzipped. Step number 3, what happens? Yes, step number 3, after unzipping, yes, now you form two strands, and these strands are separate. You see, you form those two strands. Yes, look at them. You have strand number 1 and you have strand number 2. So this strand, you see this strand, yes, and then this strand. Those strands are the one which we call templates. In science, we call them templates. In English, we call them mirrors. Don't call them mirrors. Please don't call them mirrors because we are not in English. This is not English. When you look into the mirror, you see yourself. So in science, we shall say it forms template. When you look into the mirror, you see an image. You don't see exactly the same because when you close the left, it closes the right. That's why we shall say that for formation of a complementary strand, the strand is complementary. When you say complementary, it is left and right. You understand? Left and right. So it forms a complementary strand whereby, now you tell us that whereby adenine will pair with thymine and guanine will pair with cytosine. That's exactly what happens in there. You understand? Then, if adenine pairs with adenine goes with thymine and guanine goes with cytosine. Where are these nitrogenous bases come from? Where are these nucleotides coming from? They come from the nucleoplasm. So we shall say that to form a complementary strand using free floating DNA nucleotide. Why do we should use DNA nucleotide? Because we are forming DNA. If you are so forming RNA, then it would have been using free floating RNA nucleotide. That is transcription. But here, because you are using DNA, you use using free floating DNA nucleotide from the nucleoplasm. Guys, am I clear? Do you understand what I'm saying? Yes. Using free floating DNA nucleotide from the nucleoplasm. So, meaning that now a complementary strand, yes, let me zoom it out so that you see clearly. A complementary strand, this strand, which is complementary to this old one, is formed. You understand? So now you form now two new DNAs. Oh, look, you form this DNA number one and you form DNA number two. So two new are formed, whereby, look at this critically, guys. Let me clear it up so that you see it. Whereby these two DNA, they're identical. I'm going to show you how are they identical. Each with one original strand. Look at this. Each with, look, don't just cram. Just look and then understand. If you don't understand, let me know in the comment section. Yes. Each with one original strand and, yes, one new strand. I see. It has a new strand there. And then it has an original strand. I see. Yes, it forms two new DNA, each with one original strand and one new strand. So you have a new strand there, then you have an original strand there. These two DNA are genetically identical, and this process is controlled by the enzyme. Let me show you how these two new DNA are genetically identical. So you have DNA here. I'm gonna say you have that DNA and you have that DNA. Let me say A, C, G. See, I'm not going to make it too long. A goes with T, and then C goes with G, and then C goes with G. Isn't it? Yeah. So that is the first DNA. Now I want to see that the two new DNA which are formed are identical to each other, and they are identical to the original DNA. So basically, we see that the first DNA had A, T, C, G, and then G, C, G. So that is the first DNA. Now, when they come to pair up, here I'm going to have, let me just use a different color so that we are clear. So A goes with T, and then C goes with G, and then C goes with G. And then what about here? A goes with T goes with A, C goes with G, and then mhm, C goes with G. You talk about something else, you are writing something else. Imagine. C goes with G, and then G goes with C. So basically, now let's compare. This is now the new DNA from that side. So this forms DNA 1. And then this one is another DNA from that side. So this is DNA number 2. Now let's compare. Let's see if they are identical to each other. All right. I have A here. I have A here. On the other side, I have C here. I have C here. I have C here. I have C here. So those strands are the same. What about the second strand? I have T this side, I have T this side. I have G this side, I have G this side. I have G this side, G this side. You see that this and this are the same. What about the original DNA? Let's find out. I have A here and T. I have A have C and G, have C and G, have C and G, have C and G. You see that the original DNA is exactly the same as the new DNA. What about this? I have A here, I have A here, I have T here, I have T here, I have C here, I have C here, I have G here, I have G here, I have C here, I have C here, I have G here, I have G here. It means that the two new DNA which are formed, they are genetically identical. That's what it means. So you have to know that concept. Yes. So now what do we write in exam? So basically, what we write in exam, we say DNA double helix unwind. Cool. It unwinds. When it unwinds, double helix. The word DNA double helix. Don't say DNA helix. Nope. We'll give you a zero. DNA double helix, we give you a mark. Unwind, we give you a mark. Weak hydrogen bonds break. The same thing as saying unzip. We give you a mark. Don't say hydrogen bonds break. We say weak. To form two, listen, I repeat, to form two separate, listen, I repeat for the third time or second time. To form two separate strands, don't say to form two strands. Mm-mm. We don't give a mark. This word separate is crucial. To form two separate strands. Now, because we're on DNA, we have seen that each is acting as a template. This is acting as a template. Also, this is acting as a template. So we shall say, to form two separate strands, each is this word each. If you have not used the word each, you can use the word both. Both are acting as a template. Both are acting as a template for formation or a complementary strand, for formation of a complementary strand using free floating DNA nucleotides. Those are key points. Where are they best from? In the nucleoplasm. Free DNA nucleotide from the nucleoplasm. Free DNA nucleotides are the one which are being used. So that's the key point in this regard. A complementary strand is formed whereby complementary strand is a marking point whereby adenine pairs with the thymine and the guanine pairs with cytosine. Two new DNA are formed which are genetically identical. They are formed, they are genetically identical. I've explained to you how are they genetically identical. We have seen that before and after the things are the same. And the two new DNA which have formed, they are genetically identical. They are the same. Don't say that they are identical. Mm-mm. They are genetically identical because we are talking about in terms of genes. Something can be identical, but it's not genetically identical. Identical twins, they are not genetically identical. They have some variations in their genetics, but we call them identical twins. So if you just say that they are, which are identical, you are killing yourself. You are not a distinction material yet. Say which are genetically identical. Yes. With one original strand, each with one original strand and one new. We have explained this. How is each with one original strand and how is each with one new strand? And this process is controlled by the enzyme. We don't talk about those enzymes. When you continue with your studies, you know the polymerases which are there, the helicases which are there, the ligases which are there. There are so many primases which are there. We don't talk about those enzymes. If you're using a book and it has those enzymes, please don't waste your time. No one is going to ask you about those enzymes. And if you write them, you won't get a mark. What we are looking for, this process is controlled by the enzymes, is enough. Why do you waste your time? You must not waste your time. So basically, that is the process of DNA replication. You understand? So we see that DNA double helix unwinds, weak hydrogen bonds break. We say that the word weak must be there, it breaks. And then when you say weak hydrogen bonds break, the word unzip is the same as weak hydrogen bond break. Each to form two separate strands, the word separate is a key point. Each or both are acting as a template using free DNA nucleotide. That's the key point. So using free floating DNA nucleotide from the nucleoplasm, the word free and DNA nucleotides are the key points in there. For formation of a complementary strand, understand? Yes, whereby adenine pairs with thymine and guanine pairs with cytosine. So the two new DNA are genetically the two new DNA are genetically identical. We say that don't say they are identical because genetically identical and identical is not the same. That's why the key point is on genetically identical. Genetically identical. If you miss out genetically and you just say identical, you might cross it. And don't ask why. Just follow there because I've explained it. Each with one original strand and one new strand. We have explained it here that each is one original strand and one new strand. And then we are saying that the process is controlled by the enzyme. The process is controlled by the enzyme. Yes. And we say that don't bother yourself talking about the enzymes. Sometimes the enzymes even you don't know. You understand? Yes. Yes, yeah. Some people they just want to make their life difficult too, by making the Me, I know. Me, I know. I know too much. So I want to have to write these complicated things. Woohoo. You complicate yourself. Don't complicate your life, man. All right. Let's go to some of the questions. So basically, that's DNA replication. We have some questions which we're gonna see, but these questions are more especially on protein synthesis because that is our next topic, and we're gonna cover it tomorrow. Tomorrow, guys. Tomorrow, don't miss the class for protein synthesis, and we're gonna talk about the questions. Yes, if you have a question, please let me know so that we are able to help you out. Download the distinction material so that it can help you to get a distinction. Yes. Let's just see one, two questions. We shall see these questions tomorrow. Yes, because they are about protein synthesis. But before we go to that one, let's just talk about this question. Where are you? I think I didn't add it there. Ne? Yes. I didn't add it there. Yeah. Mm, yeah, I'm going to stop here. So we will do some questions tomorrow once everything is done. But before I go to the end of the class, guys, remind me, what is DNA replication? DNA replication is the process by which DNA makes identical copies of itself. When does it occur? It occurs when. When you say when, what are they? They're talking about when does it occur? It occurs before the cell divides or during interphase. Remember, interphase is not phase of meiosis. We shall see that when you go to meiosis. It's not phase of meiosis. No, it's just a growth phase. We shall see it. So, why does it occur to double the chromosome number? Where does it occur? It occurs in the nucleus. How does it occur? It occurs, you explain how does it occur by explaining DNA replication. Explain DNA replication. Then you'll be able to understand how does it occur? So these questions, guys, make sure that before you go to the paper, you answer these questions. DNA replication doesn't have many questions. You just have to know these questions I listed here. then you'll be able to understand and answer all questions concerning about DNA replication. What are the building blocks of DNA? Remind me. We say that they are nucleotides. What are the monomers of DNA? There are nucleotides. You understand? What are the units of the nucleotides? Yes, sometimes they can ask you that question. What are the units of nucleotide? Remember, nucleotide is looks like that. Eh? Looks like that. What are the units of nucleotide? It's the phosphate group, the sugar, and the nitrogenous base. So nitrogenous base, so these are the units of the nucleotide. If they ask you what are the units of DNA nucleotide, then you talk about the phosphate sugar, a deoxyribose sugar. In this case, you talk about a deoxyribose. Yes, you're talking about deoxyribose. And deoxyribose, because deoxyribose, you are talking about DNA, deoxyribose sugar. And then the another question they can ask you, then nitrogenous base. In this case, you talk about adenine, thymine, guanine, and cytosine if they're talking about DNA. Guys, I think I'm clear. There's a student who asked me a question. Let me check here. Um, ask me a question. Um, I can't see the question. Let me check. Okay. I think you were asking me questions about DNA. If you have a question, let me know. If you don't have a question, guys, we're going to stop here. We shall meet again tomorrow. Yes. Don't forget to subscribe, like, and also have some subscription to support us, and then we also support you. Those subscription they help us so that we can also prepare content for you. Eh? Yes. See you again. I think tomorrow. Tomorrow I should be live with the face. Ne? Who wants to be? Who wants me to be live? Just to see me when I'm teaching live. Ne? Not just live with the screen. Live when you see me. When I ask you a question, you ask me. You answer me. Ne? Let me know in the comment section. Ne? If you want, say you just type there live, 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 live. Ne? See you guys. Ne? See you guys next time tomorrow. Don't miss out.